Good morning, good afternoon or good evening from GFoRSS TV.
Good morning, good afternoon, good evening, wherever you are around the world, and welcome to this new broadcast of GFoRSS TV. Today's broadcast is dedicated to reviewing changes in the food regulatory framework of a major market that is the European Union, and to understand the repercussions of this change on some aspects of food regulatory science. And we are speaking here about food analytical science.
You are watching GFoRSS TV. Today, on GFoRSS TV.
In March of 2021, the European Commission published a regulation known as 2021/808. This regulation is related to the performance of analytical methods for residues of pharmacologically active substances used in food-producing animals. This regulatory requirement will be implemented and will be enforced in the coming months. We are fortunate to have with us experts from the Waters Corporation who will be explaining the repercussions of this change in regulations on the way analytical methods are meant to be developed, verified, and applied. We wish you a great broadcast.
Coming up next, on GFoRSS TV.
Hi, my name's Simon Hurd from Waters, and we're going to talk to you today about the Commission implementing Regulation 2021/808. And we're going to introduce the session with a refresh on the regulations in Europe associated with vet drug residues. So the presentation today is going to start with a brief introduction. We're then going to talk a little bit about the different regulatory limits and control of banned and authorized substances within the European Union. We're going to talk about the European Union regulations for official control, and we're going to look at the residue requirements for non-EU countries exporting into the European Union, and then we'll summarize at the end. Vet drug products are used for two purposes, in the main to prevent and control disease in animals, but also for growth promotion as well. These substances unfortunately may leave residues in the food from treated animals. Animals can accumulate all sorts of different chemicals in their tissues. So for example, we can have naturally occurring toxins like mycotoxins. We can have environmental contaminations such as dioxins, and we can even have various contaminants introduced via feed. In all cases, the level of residues in the food should not harm the consumer. So before a drug is authorized and placed on the market, its safety must be evaluated. And that procedure looks at the residues that are found in the tissues and recommends maximum residue limits. The MRL is the maximum concentration of residue resulting from the use of a veterinary medicine product, which may be accepted to be legally permitted or recognized as acceptable in or on a food. MRLs can be specific or generic. So for example, we might have the same value of an MRL for a substance in all food producing species, or that substance may have different MRLs for different species. So the MRL for beef may not be the same as their MRL for pork. We also have different MRLs for different types of tissues. So for example, the MRL in muscle will be different to the MRL in kidney or the MRL in liver. We also have some MRLs for products of animal origin, such as for honey or for milk. It is important to note that if there is no MRL, unlike for pesticides, we have no default LOQ LMRL system. Some of the key regulations associated with MRLs are laid out in this slide, so you can actually access the links later on. This particular regulation, Commission Regulation 37/2010, is the critical one. It actually has all the MRLs actually set out in the annex. However, remember that this regulation has been amended many, many times, so make sure that you're looking at the most recent amendment. So this is an example of an entry in the annex which gives us the MRLs for two substances, two antibiotics. So if we take the first row, we can see the compound name, and we also see the marker residue, and I'll explain what that means in subsequent slides. We can see that this particular product is approved for 3 different species, bovine, porcine, and horse, and it has different MRLs for the different tissue types. And we can see it also has an MRL for one of the food producing products, milk. On the second row, you can see a second compound, the different type of marker residue. So we'll come back to that in the subsequent slides. In this particular case, it's approved for all mammalian food producing species. But again, the MRLs differ quite considerably between the different animal tissues and, in this case, milk. During the evaluation of safety that takes place before a product is authorized, as I said, the residues in various animal tissues and food are determined. But in some cases, it's no longer the parent vet drug. It actually may be that in the animal that the vet drug is metabolized. So it's important to understand the metabolism, which is this type of study is performed in the target or the food animal species, and also at this stage a marker residue is determined for regulatory monitoring purposes. It is important that this marker is suitable for monitoring, so it has to be stable, it has to be quantifiable with readily available analytical technology, and it should be representative of the total residue concentration. So for most vet drugs, the marker residue will be probably the parent compound or a metabolite or some combination of thereof. However, this is often expressed in quite different ways. So if we take, for example, tetracycline, it's got quite a simple marker residue. It is the sum of the parent drug and its 4-epimer. However, the second compound here, albendazole, is actually the sum of albendazole metabolites, as you can see, expressed as albendazole. Amitraz is a little bit more complicated. It's the sum of amitraz and any metabolite containing a specific moiety associated with the parent compound. And then lastly, an example here of actually a marker residue that actually requires a hydrolysis of all of the metabolites back to a specific product. One important point to note is that MRLs are established only for those food-producing animals for which an application is made. If a substance that can be used as a vet drug is not listed, this means that vet drugs containing that substance cannot be authorized for use. So therefore, residues of this substance should be absent in tissues or products of all food-producing animals. For example, we have MRLs for residues of ivermectin in target tissues of bovine and ovine, but we have no MRLs in milk, poultry, pigs, or other products from other food-producing animals. So this means that veterinary drug products that contain ivermectin cannot be authorized for use in, for example, poultry. So we should not ever see residues of ivermectin in tissues of poultry. The European Union operates a zero tolerance policy to residues of prohibited substances. But what does this mean to analytical testing? Prohibited substances have no MRLs. So what limits do we use to actually enforce zero? So some years ago, to ensure laboratory performance was consistent, minimum required performance limits, MRPLs, were set up for a limited number of banned substances, for example, chloramphenicol, nitrofurans, malachite green, leucomalachite green. The MRPLs were the minimum content of an analyte in a sample which at least has to be detected and confirmed. They were not legal limits. Please note though that the term MRPL has now been discontinued and replaced. A new system, reference points for action RPAs, have been established for prohibited substances when it is deemed necessary to ensure the functioning of controls in food of animal origin imported or placed on the market. If you want to know more information about the reference points of actions, and you click on the link and that'll take you to the European Union website. I've given you here some examples. Well, actually, these are the updated reference points of action. We have reference points of action for chloramphenicol, 0.15 micrograms per kilogram, malachite green and leucomalachite green. The sum is 0.5 microgram per kilogram. And then we have nitrofurans and their metabolites, or as their metabolites, 0.5 micrograms per kilogram for each of these metabolites listed. So straight away you can see that the numbers are very small. There's actually half the previous MRPL values, but also you can see here there's an additional nitrofuran that's been added to the list as well. These new reference points of actions shall apply from the 28th of November 2022. As you can see from the previous slide, the number of reference points for action for banned substances is actually still really small. So what do we do about other banned substances? So in 2020, the term Minimum Method Performance Requirements, MMPR, appeared in a guidance document to replace the original recommended concentrations. For banned substances in food though, please first just check that there isn't a reference point of action. These values in this document, the as regulatory limits, a very important point to stress. They're used to indicate the sort of analytical performance in the different types of matrix. So it's a really helpful guidance document to help us set up validation for banned substances where no RPA exists. The key regulation associated with official controls within the European Union is now Commission Regulation 2017-625, as it lays down rules for the performance of official controls and other official activities by competent authorities within the member states, and of course also imports into the European Union as well. It provides specific rules on official control in relationships to all substances whose use may result in the residues in food and feed. One of the critical things to note is it provides an obligation for member states to ensure that official controls are performed on the basis of a multi-annual national control plan. We'll talk about that in a minute. It also talks about obligations for each member state as regard to actions to be taken in case of suspicious or non-compliant situations. In addition to this regulation, there are numerous additional implementing measures and supporting controls and delegated acts which have had to have been adopted to replace those previously linked with earlier controls. So this schematic shows how it integrates the controls covering food and feed laws, rules on animal health and welfare, plant health and plant protection products. Here on this particular diagram, we're just really talking about the chemical food safety, but you can see that the regulation now covers various contaminants, food contact materials, residues of vet drugs, substances having hormones effects, and pesticides, all in a single regulation. So as I stated on the previous slide, European Union countries must implement residue monitoring plans to detect the illegal use or misuse of authorised veterinary medicines in food-producing animals, and to actually investigate to follow up the reasons for those residue violations. The monitoring programs take place every year, and the results are reported through the EFSA website. Some member states actually also run additional surveillance programs for other purposes, depending on their needs. The regulation classifies the veterinary drugs into different substances and categorizes them as two groups, A and B. The A group contains most of the substances banned for use in food-producing animals in the European Union, and it is further subdivided. The second group contains residues of the pharmaceutically active substance present in the vet drugs, which may be authorized for use in food-producing animals. Group B, interestingly, also contains pesticides and heavy metals. And again, Group B is considerably subdivided. Please note, though, that there are plans to change these groups, so you need to keep an eye on the regulations coming out. As I said previously, the results from the national residue monitoring plans across Europe are published every year, and the latest document is actually from the 2020 programme. And you can see from the slide that numbers of targeted samples reported for residues of specific veterinary medicine products. And you can see that's a very, very low non-compliant rate. So this is not detection or quantification. This is looking at substances that are deemed, or residues rather, that are deemed to be non-compliant. So it's less than 0.3% of the samples. So this level of non-compliance is so low that it actually drives a two-tiered approach to the analysis: screening and then confirmation. So I'd like to briefly just draw attention to a couple of other changes in the regulations on monitoring. So this Commission delegated regulation 2022-931 lays down rules for performance of official controls as regard contaminants in food. And what it's doing is basically saying what type of commodity groups need to be tested for what substances. It also contains criteria for sampling strategy as well. The second major change in regulations is the Commission implementing regulation 2022-932. This basically talks about the monitoring programs actually and makes quite a lot of changes to the structure. So now the monitoring program should contain the following plans: a national risk-based control plan for the production in the member states, a national surveillance plan for production in the member states, and a national risk-based control plan for third country imports. There are specific residue requirements for non-European Union countries that export into the European Union. So these European Union rules affect trading partners worldwide. So non-European Union countries wanting to export to the European Union must also ensure the same level of safety as that of the European Union itself. The requirements for these trading partners are captured in this very helpful manual. But the two things to note: firstly is the amount of testing that goes on at border control posts to ensure that imports meet the regulatory requirements. And then secondly, that third countries wishing to export food of animal origin to the European Union are required to submit to the Commission services an annual residue monitoring plan of their own for the commodities in question. And then the third aspect is the audits that are taking place to ensure that those plans are implemented correctly. These national plans are supported by increased levels of import controls and extra safeguard measures. So in situations where the monitoring program is showing a significant risk from certain commodities or certain substances in certain commodities from certain countries, then additional regulations are implemented. So for example, here we have a specific regulation associated with India on emergency measures applicable to consignments of aquaculture products imported and intended for human consumption. So this will target a specific commodity, say for example looking at nitrofurans, and also say how much of the imports have to be tested. The other area which is very important is the fact that monitoring is carried out by or for the food industry, food business operators on exports in the third countries themselves. And this is a really important aspect. It's very cost effective to try to solve residue problems in-house within the country before they get shipped halfway around the world. So again, in India, there are significant amount of pre-harvest and pre-export testing of aquaculture products. One major tool that's used within the European Union to monitor the safety aspects of imported products, imported food and feed, is the Rapid Alert System for Food and Feed, RASIF. So this system, set up a number of years ago, enables information to be shared efficiently between members and across Europe to ensure that urgent notifications are sent, received, and responded to collectively. So if we have a risk that's detected within a certain country within Europe, one of the member states, then that country will share that information with the rest of the European Union so that the situation can be controlled more effectively. So if we look at total number of border rejections in 2021 for all sorts of stuff, we've got 1,469 notifications. Actual fact, if you look at vet drugs themselves, there are only 35 notifications, and actually only 4 were very serious, i.e. border rejections. And you can see here the types of issues that were currently or certainly applicable in 2021. So we had an issue with chloramphenicol in port casing from China. Nitrofurands, of course, still an issue, but much less so, as we'll see in the next slide, and then chloramphenicol as well. If we look over a longer period of time, and we look at the RASIV notifications for residues of veterinary drugs, so if we go all the way back to 2011 up to the present day, we can see that the numbers of notifications are dropping every year. And this is an indication of improved controls, not only within the European Union, but also within the countries that are exporting the products as well. And we can also see the sorts of issues that we've had to deal with. So chloramphenicol nitrofuran metabolites, a significant issue 10 years ago. And then things like chloramphenicol in enzyme preparations, and then more recently fipronil in poultry, which was more of an internal issue within the European Union than imports from outside. So one of the most critical points is that if we're going to carry out all this analytical testing, the results really should be reliable. It doesn't matter where you are in the world, who you work for, you should be able to compare the results and actually have confidence in those results generated. And this is a vital requirement for the Commission's approval of residue monitoring plans and listings for third country as eligible to export products of animal origin intended for human consumption. So those laboratories carrying out the testing need to demonstrate their analytical methods are fit for purpose, that they are capable of accurately determining residues at the level of interest, whether that be an MRL compound or a banned substance, and they are reliable, i.e. they perform consistently. Laboratories must use only analytical methods that are validated, and we're going to talk about that in subsequent modules, and demonstrated to work in the laboratory in a reliable way. And also, very, very important, laboratories must have a documented quality assurance system in place which will demonstrate that methods perform reliably over time. And so ISO 17025 is really important in this case. And we can see from this slide how important ISO 17025 is now. It is called out in the regulation 2017-625, and also the Commission approval of a third country residue monitoring plan is contingent on the competent authority in that third country using an ISO 17025 accredited laboratory. So I'm now going to give a brief introduction to Commission implementing Regulation 2021-808. We'll be spending a lot more time on this particular regulation in subsequent modules. This regulation lays down rules concerning the methods used for sampling and for laboratory analysis in relationship to residues of vet drugs. It's applied to official controls aimed at verifying compliance with the requirements on the presence of vet drugs. It includes details about validation so that results can be compared, and it also lays down rules for interpretation of analytical results of these laboratory analyses. And it replaces the earlier decisions that you can see on the slide, which are now repealed. So we've talked a lot about compliance and non-compliant samples, but how do we know whether a sample is compliant or not? It is essential to have some means to interpret the legality of any analytical results in a consistent manner if we really want to compare and contrast between different laboratories. So the result of an analysis should be considered non-compliant where it is equal to or above the decision limit CC alpha, not actually the MRL. The alpha error is the risk of erroneously rejecting a correct result. So CC alpha is all about avoiding false non-compliant decisions. So what does that mean? For example, even though the concentration is truly compliant, the result is actually reported as non-compliant. CC alpha values are not used to estimate the minimum amount that the method can detect, but for simply deciding whether the result is compliant or not. So this type of diagram is used a lot to explain CC alpha for MRL substances. So what we have is a representation of multiple measurements of a sample or many samples at the MRL. So basically, if we had a fantastic, in an ideal world, we would basically have measurements that would give us back the concentration at the MRL every single time, and we would just see this line here. But of course, we have some uncertainty around our measurements, and so we end up with this profile of actual results. So what we're doing with this type of approach is basically saying that we want to assess the data not based upon whether the measured value is greater or less than the MRL, but actual fact whether it is greater or less than the MRL plus the error of the method. So this is where we actually include this factor with the error around the repeatability of the method, and then we generate a new value called CC alpha. And basically that means that we're no longer assessing the compliance of our data against a limit, regulatory limit, but we're assessing the compliance against the CC alpha that's been generated by that laboratory. Another way of looking at the relationship between the MRL and the CC alpha for confirmation is shown in this diagram. So we can see here the compliance versus non-compliance in terms of concentration, and then we can see the MRL, and then we can see the CC alpha, which is obviously the MRL plus the uncertainty associated with the method. So CC alpha is the MRL plus the error associated with the method. For MRL substances, the CC alpha should be higher than, but as close as possible to the MRL. When it comes to banned or unauthorized substances, the same process is applied, but in a slightly different way. So here we have zero tolerance, so we don't have a legal limit. In an ideal world, we should have zero. So again, in theory, then of course, if we're not going to be able to measure zero every single time, we're going to have this Gaussian distribution of measurements. And basically, again, instead of having a 95% confidence limit in here, we want a 99% confidence limit. So the alpha is actually 1 instead of 5%. And so again, we have the 0 plus the error associated with the method, which gets us to our CC alpha. In this case of banned and unauthorized substances, CC alpha should be as low as possible and certainly less or equal to any RPA established. So now we've determined that we use CC alpha to interpret the results to see whether our sample is compliant or not. But how do we actually create these values? So CC alpha values are calculated during validation of confirmatory methods, and we're going to talk about this a lot more in the next modules. It's also to note that we need analytical methods, which it can be demonstrated in a documented, traceable manner that they are actually validated to comply with these needs. So for example, if we're talking about prohibited or unauthorized substances, they have to be seen to have a false non-compliant rate, an alpha error, which is less than or equal to 1. And for MRL substances, that alpha error can be less than or equal to 5%. So to summarize, food producing animals may be treated with veterinary medicines to prevent or cure disease. These substances, unfortunately, may leave residues in the food from treated animals. The food also may contain residues of other materials such as pesticides or natural contaminants. In all cases, though, the level of residues in the food should not harm the consumer, and that is why we have a legal framework to actually establish this before a product is put on the market, and also then to actually monitor the use of that product. In terms of monitoring, European Union countries must implement a residue monitoring plan to detect either the illegal use or the misuse of authorized vet drugs in food producing animals, and they also must then investigate the reasons for those residues violations. But certainly within the European Union, the frequency of non-compliance is extremely low. Non-European Union countries exporting to the EU must also implement a residue monitoring plan which guarantees an equivalent level of food safety. It's good to know that the frequency of alerts through the RASIF system for vet drug residues has certainly declined over the last 20 years. So the system is working. There is a transition period from implementing the new regulations associated with official control, including imports, and that ends at December 2022. And then Commission implementing Regulation 2021-808 is the new regulation that lays down rules concerning the method of analysis used for sampling and for laboratory analysis. And again, we're going to go into a lot more detail in the next module. So I hope you found that refresh of the European Union regulations useful, and I look forward to catching up with you another time. Thank you.
Coming up next on GFoRSS TV.
My name is Simon Hurd from Waters, and welcome to the second part of the discussions around Commission implementing Regulation 2021-808. In this module, we're going to have a look at an overview of the changes from the original Commission decision 2002-657. So we're going to start off this presentation by introducing you to Commission implementing Regulation 2021-808 in a little bit more detail. We're going to talk about what's called the level of interest and then how we interpret the results before we move on to the requirements for confirmatory methods and then a summary. The regulation lays down rules concerning the methods used for both sampling and for laboratory analyses in relationship to residues of vet drugs in live food producing animals, their body parts, fluids, etc., as you can read on the slide. It applies to official controls aimed at verifying compliance with the regulations on veterinary drug residues. It includes details about how to validate analytical methods so results can be compared, and also some ongoing analytical control, quality control requirements. It lays down the rules for interpretation of analytical results of these laboratory analyses, and it replaces these earlier decisions. And here really we're going to focus on the differences between the earlier decision 2002-657 and the new Commission implementing Regulation 2001-808. There's also a second Commission implementing Regulation 2021-810, and this is where we find the information regarding the transitional provisions from the earlier decision to the current regulation. And there are two areas to look at. The first involves looking at performance criteria and validation in the earlier decision, and we have until June 2026 to move across to the new requirements. And the second relates to the migration from the MRPLs to the RPA values, and that needs to be done by November 2022. So the contents of the new regulation actually split into two different parts. There's what I've called the legal part, which is expressed in 8 different articles. And the most important parts of this are probably the method of analysis, quality control, and interpretation of results. It's also important to note now that this regulation also includes the information on methods of sampling, which was previously found in this decision 98-179. And then the second part of the regulation is basically the technical annexes. And this is basically gives us the details. So we have basically a lot of details about methods requirements. So we have a chapter talking about method criteria and other requirements for analytical methods, a whole chapter on validation, chapter on quality control during routine analysis, including a relatively new concept for this regulation called ongoing method performance verification, and then information about how you extend the scope of validation of a previously validated method. And then the second annex talks about sampling procedures and official sample treatment. Now, although this document was devised by the European Union's Commission, the European Union reference laboratories had a considerable contribution to make, and that contribution has not stopped. What we have is now a series of very, very useful European Union reference laboratories guidance documents. So we have a document on quality control, on confirmation methods, on the extension of methods, validation of screening methods, and on high resolution methods. So defining the level of interest. The level of interest means the concentration of a substance in a sample that is significant to determine its compliance with the legislation. And we have two groups. We have substances with MRLs, and we also have prohibited or unauthorized substances. And we deal with the latter using reference points of action where available. However, the level of interest, as we'll see, also impacts the design of the validation protocol. Again, we have the same thing. We have substances with MRLs, and we have prohibited or unauthorized substances. For prohibited or unauthorized substances, the document talks about using reference points of action. But in the absence of any values for RPAs, it also talks about a concept called lowest calibrated level. We also have a separate guidance document which gives us values for some banned substances, minimum method performance requirements. And this is a very useful tool to give us the concentrations that we might validate these compounds at. So it's just a quick reminder about the definition of the interpretation of results. So the result of a confirmatory analysis shall be considered non-compliant where it is equal to or above the decision limit for confirmation, i.e. the CC alpha value. For MRL compounds, the CC alpha shall be the concentration at or above and above which it can be decided with a statistical certainty of 1 minus alpha, i.e. 95% sure that the permitted limit has been exceeded. For prohibited or unauthorized substances, the CC alpha shall be the lowest concentration level at which it can be decided with a statistical certainty of 99% that that particular analyte is present. This figure shows the distribution results from the measurement of a sample at the MRL. And we can see from the diagram that our measurements are always associated with some sort of error. And CC alpha is calculated by adding this error to the MRL. When we look at the results from the sample, we no longer just assess the results or the compliance of the sample against the MRL. We assess the compliance of the sample against the CC alpha value generated at validation. And CC alpha for MRL substances shall be higher than, but as close to possible as the MRL. For banned substances, the situation is similar, but instead of having an MRL, of course, this is zero tolerance, so we're supposed to be working at 0. So we have a similar situation where we have to consider the error of the methodology. Now, in this particular case, for banned substances, CC alpha should be as low as possible and certainly less than or equal to any RPA established. So let's look at the requirements for confirmatory analysis. So in an ideal world, confirmatory methods should provide information on the structural chemical composition of the analyte. For prohibited and unauthorized substances, mass spectrometry is essential. For those with MRLs, there are some exceptions. So if the methodology is suitably validated, a method with diode array detection or fluorescence detection may prove suitable. A method that relies just on single wavelength UV will not be. Before we go into detail about some of the general performance criteria for confirmatory methods in the document, in the regulation, I thought it important to define some of the terms. So for the rest of this presentation, I'm defining accuracy as the closeness of agreement between a test result and the accepted true reference value. And this is determined by estimating trueness and precision. And we can see an illustration of this in the slide. The regulation defines trueness as the closeness of agreement between the average value obtained from a large series of test results and an accepted reference value. And the new regulation uses a similar approach to the previous decision, where trueness can be calculated based upon spike samples as an alternative to using certified reference materials. Unfortunately, in the vet drug world, it's very rare to find any certified reference materials that might be available. The regulation gives you a calculation that you can do for trueness, and it actually explains that we have to calculate this trueness value for each spiking level at validation. Precision is the closeness of agreement between independent test results obtained under predetermined conditions, and it is expressed as the relative standard deviation or coefficient of variation of the test result. And the new regulation differentiates between precision under repeatability and under reproducibility conditions. Some more important definitions. So repeatability, according to the regulation, is the closeness of agreement between independent test results obtained under the same conditions, whereas within laboratory reproducibility is the closeness between independent test results obtained under different conditions in a specific laboratory. So most commonly, this may refer to different instruments, different operators, but also different days. So you might repeat the experiments over a period of 3 days. This is also called intermediate precision or in-house reproducibility. According to the regulation, reproducibility conditions are where test results are obtained with the same method on identical test items, but in different laboratories. And this is really more commonly known as interlaboratory reproducibility, to make it clear from the within laboratory reproducibility. This slide shows us the minimum trueness for quantitative methods, and we can see that the acceptable range varies depending on the concentration in the sample. And the only real change is to the range acceptable for residues greater or equal to 10 micrograms per kilogram. This slide shows the range of values that are acceptable for within laboratory reproducibility, and we can see that they change depending on the concentration. At low concentrations, the Horrocks equation doesn't do a very good job of estimating what should happen. So therefore, the within laboratory reproducibility really needs to be as low as reasonably possible. And then also note that repeatability conditions, under repeatability conditions, shall be equal or to below 2/3 of the table values. So when it comes to chromatographic requirements, the first thing we need to note is how much retention do we need? And the way it's expressed is as the minimum acceptable retention time for the analytes under examination should be at least twice the retention time corresponding to the void volume of the column. So if we look at this chromatogram here, we see a peak at 2.49 minutes. And we look at that and we think, well, that's not bad. That's quite a nice retention. But we need to actually have a look at the size of the column. This is a pretty big column, a wide column and a long column. So what's the actual void volume of that column? It's actually 1.74 mils. So at a flow rate of 0.75 mils a minute, the time equivalent to the void volume is 2.33 minutes. So in reality, we have very little retention, if anything at all. And so if the peak is unretained, the likelihood is it's going to collude with a whole load of other co-extractives. Retention time is also an important identification criteria. So the retention time of the analyte in the extract shall correspond to that of the reference standard within a tolerance of plus or minus 0.1 minute, or when the retention time is below 2 minutes, a deviation of less than 5% of the retention time is used. And there are slightly separate requirements for when an internal standard is used. And we can see from the graphic what sort of tolerances are acceptable. The document talks about calibration. So it gives us information about calibration graphs when they're used for quantitation. For example, it specifies that at least 5 levels, including 0, should be used for construction of the curve. It also talks about the parameters of the curve that need to be described. So for example, the working range of the curve, the mathematical formula of the curve, and the goodness of fit of the data to the curve all need to be reported. The calibration graph should cover the range of interest. This is a really important point. If you've got a calibration graph like this, you need to be making sure that the calibration graph is suitable for your measurements. So if your measurements are within the sort of range of the calibration graph, then everything's fine. But we should not be trying to extrapolate beyond the calibration graph because we don't know it's linear, and we certainly shouldn't be extrapolating lower in the calibration graph. Interestingly, the document doesn't provide any fixed criteria such as values for R² or values for residuals. And interestingly, there are different approaches to preparing standards for calibration, and that's what we're going to talk about next. The first type of calibration standard is based in solvent standards. So this is a series of analytes prepared in solvent at different concentrations. I would only recommend the use of solvent standards when you've investigated that there is no significant matrix effect. The second very common type of calibration standard is what we call matrix matched. So this is a series of analytical standards prepared instead of in solvent in a blank extract of the matrix being tested. And this does very well to mitigate for changes in response due to matrix effects. The third type of standard calibrant I want to talk about is called matrix fortified. It's also called matrix extracted or procedural standards. And the matrix fortified standard is a series of analyte standards prepared in a blank test portion of the matrix being tested. So it's actually using the same procedure as we would use to spike a blank. However, in this case, it's used for the primary calibration. The use of matrix fortified standards mitigates for any changes in response due to matrix effects, any losses during extraction and cleanup for recovery correction, and there's no need to calculate absolute recovery at validation using this approach. One common option used for calibration for vet drug testing is the use of internal standards. An internal standard is a substance that is not contained in the sample and has a physiochemical property as similar as possible to the analyte. So, for example, stable isotope analogues are a classic case. Stable isotope analogues or any other internal standard can be added at two stages. We can carry out the extraction, any other cleanup, any other prep work, and then actually add the internal standards right at the end prior to the LCMS-MS. If we do that, it corrects for any matrix effects and it adds to the precision or improves the precision of the measurement. And because we're adding it at the end, the cost is reduced because we only have to add a small amount. The alternative, and probably from a vet drug perspective, more common use of internal standards is to add them right at the beginning during the extraction, prior to extraction. And internal standards here actually corrects for recovery and improves method precision. And there's no need to calculate absolute recovery at validation because you have already corrected for recovery. It also, of course, corrects for any matrix effects as well. The downside is that because you have to add the internal standards at the beginning, you have to have a greater concentration, which obviously adds to the cost. When it comes to using mass spectrometry for identification and the associated requirements, the use of ion ratio data is extremely common when we use a tandem quad platform. And here the regulation specifies an ion ratio of plus or minus 40%. Now this is quite a change from the previous decision where there was a sliding scale. So I'm going to show you some chloramphenicol data as an example. So here on the left-hand side we have an injection of a standard, and we can see the two transitions for chloramphenicol, 321 to 152 and 321 to 257. And we have the peak areas of the two transitions. And all we do is to ratio them. And if we do that, we get a ratio of 0.69. However, it's probably better practice to calculate the mean of the ratios from all of the calibrants that you have run within the same batch. If we then look at a sample, again, same transitions, and we use the same thing, we get a peak areas and we can calculate a ratio of 0.71. So we can see the comparison of the sample to the standards, very similar, well within the 40% of the tolerance in the regulation. The other big change in this new regulation is the introduction of the use of high resolution mass spectrometry. So we are now talking about time of flight mass spectrometers or Orbitrap instruments, and they are now included as appropriate for identification purposes. So previously it was possible to utilize these instruments for screening purposes, but not for identification. So this new regulation talks about confirmation of identity of an analyte. Using high resolution mass spectrometry, you need a mass deviation of all diagnostic ions below 5 ppm, and then there's another limit for smaller masses. On the basis of this, the resolution should be selected fit for purpose, and the resolution should typically be greater than 20,000 at full width and half maximum. And the document does provide some guidance on the selection of diagnostic ions. The other major change to the new regulation is changes to the identification point system. So the table on the right hand side shows the number of identification points that each of the techniques yields. And it's interesting to note that now we have separation called out as a separate identification point. For high resolution mass spectrometry, the identification points will be dependent on the mode of acquisition used. So for substances for which an MRL is established, authorized use, a minimum of 4 identification points is required, whereas for banned substances, we need 5 identification points. And I'm now going to take you through an example using tandem mass spectrometry. So if we want to confirm a sample and we want to meet the identification point rules for a banned substance, if we use LCMS-MS and we have 2 transitions, all our criteria, of course, are met, we get 1 identification point for the chromatography, we get 1 identification point for the precursor ion, and then we get 1.5 points for the product ions. So the sum of that equals 5, which is within the requirement both for banned substances and for MRL substances. So the take-home message is that when you're using LCMS-MS on a tandem quad or even on a high-resolution mass spectrometer, as long as you're doing MS-MS, if you use 2 transitions for ion ratios and the retention time is sufficient, retention time tolerance is met, then this is sufficient for both band and MRL compounds. So let's summarize some of these changes for performance characteristics. So if we just focus just on the identification points for a second, for example, we have now for iron ratios, they are plus or minus 40% across the board. And we now have the introduction of the use of high resolution mass spectrometry definition for mass accuracy and resolution, and also the introduction of a slightly different identification point system where we have this extra identification point for chromatographic separation, but we have the need for increasing the number of identification points to meet the criteria 4 and 5. For chromatography, generally the requirements for validation are the same. Requirements for identification, though, have been adjusted. So now we have an absolute retention time criteria, and we have a slightly different permitted relative retention time deviation reduced to 1%. So now I want to move on to talking about validation. So method validation is the process of proving that an analytical method is acceptable for its intended use. Validation is a requirement of ISO 17025, and validation of a method is typically undertaken before that method is implemented for routine analysis. The approach taken in the new regulation is very similar actually to the previous decision, but there are some small changes. The performance characteristics that need to be verified vary on the type of testing required. So for example, screening versus confirmation. And here today, we're just going to focus on confirmation. And Commission Implementing Regulation 2021-808 focuses on a single laboratory in-house validation. So when considering the design of a study plan in this new regulation, the legal status of a substance needs to be considered as this determines the spiking concentration. So for an authorized substance, the MRL is the reference for any validation for the spiking. Regarding banned substances, unauthorized substances, we use the reference point of action where it exists. Where no RPA value exists, the regulation specifies substances need to be validated based upon what they call the lowest calibrated level, meaning they should be validated for concentrations which are as low as reasonably achievable. Although the minimum method performance requirement is not called out in the regulation, these values that are being provided by the European Union reference laboratories are useful to help us set up validation experiments for these banned substances where no RPA exists. As per the previous decision, Commission Implementing Regulation 2021-808 lays down required spiking levels for validation. If we look at an authorized substance, that substance with an MRL, then the MRL itself is the reference point for the spiking. So we have spiking at 1 times the MRL and spiking at 1.5 times the MRL. The lower limit used to be 0.5 times the MRL, but in this new regulation it has been changed to 0.1 times the MRL. However, please note that where validation at this low level is not achievable, the regulation does allow you to use a concentration between 0.1 and 0.5 times the MRL if it is more reasonably achievable. In the second case, we have the banned substances or unauthorized substances that have a reference point of action value, and we use the reference point of action as the reference. So here we haven't really changed from the original decision to the new regulation. We have 3 spiking levels, 0.5, 1, and 1.5 times the reference point of action. In the third case, we have unauthorized substances, banned substances that have no RPA. And this is a new concept that's introduced within this regulation, and that is the lowest calibrated level. The good news is that we can use the MNPL value as the reference point for spiking. These MNPL values are provided by the European Union Reference Labs guidance document. And lastly, but not least, at each level, the analysis needs to be performed with 6 replicates. One of the challenges associated with validation exercises is that there is a lot of calculations required. Identification criteria such as retention times or iron ratios can be evaluated using instrument vendor software. After analysis though, concentrations are used to calculate the performance characteristics, and this is best done using statistical software such as Excel or in various bespoke packages that are available. Calculations can be based on the analysis of variance and over approach. to determine trueness, repeatability, and within laboratory reproducibility. And we will discuss this in more detail in the next module. There are various other ways that you can do the calculations which are listed in the regulation. However, please note that any software or spreadsheets required for these calculations need to fulfill the criteria of ISO 17025. So this slide gives an overview of the changes in the requirements for method performance characteristics from the original decision to the new regulation. So we can see from identification that the change in the concept for identification points is one of the key things to look at. So this is, for example, the addition of the 1 point for identification point for chromatography, but also the introduction of high resolution mass spectrometry is another important change. From a point of view of chromatography, the general requirements for validation remain the same, but requirements for identification have been adjusted slightly. There have been no changes in the requirements for calibration curve. The concentration levels that are required for the spiking have been changed especially with the MRL-based substances. The precision and the trueness values have been adjusted a little bit. Measurement uncertainty, which we've not really talked about here, is actually called out in this regulation, but was not explicitly mentioned in the previous decision. Relative matrix effects, again, is a new concept in this new document, and absolute recovery previously was referred to as recovery. There have been no changes in the requirements for specificity, selectivity, or ruggedness. From the point of view of the CC alpha, there are some changes to the calculation methods. In fact, there's an additional calculation method available. One of the biggest changes is the concept of CC beta. This is no longer required for confirmatory analysis, and now is solely the purpose of CC beta is to establish the performance of a screening method. And lastly, but not least, there have been no changes in the requirements for stability testing. In summary, Commission implementing Regulation 2021-808 lays down rules for the analytical methods and sampling to be used in the testing for residues in live animals and animal products. It replaces the previous decision. There is a second implementing regulation that covers the transitional period provisions for certain substances. The new regulation is still designed to distinguish between compliant and non-compliant samples in official control. It specifies common criteria for the interpretation of analytical results. And one of the new areas is the identification point system now allows for the use of accurate mass measurements from high resolution mass spectrometers. There are some new features such as absolute recovery and the concept of relative matrix effects. The level of spiking levels for validation still depend on the class of substance and the level of interest. The lowest level for the MRL substances has been changed from 0.5 times the MRL to 0.1 times the MRL. The regulation includes updated information on choices of methods, analytical quality control, validation, and sampling. It is intended as mandatory for official control purposes. However, the regulations are still rather complicated, especially when you compare them to the validation requirements required for pesticide residue analysis. And this is where these guidance documents provided by the European Union reference laboratories are so useful. And that brings us to the end of this module. I hope you enjoyed it, and I look forward to talking to you again soon.
You are watching GFoRSS TV. Today, on GFoRSS TV.
Through this broadcast, we have once again emphasized the importance of the fact that regulations are equivalent to living organisms, in the sense that they are not meant to be static, they are meant to evolve to change, and to adapt. Food regulatory science are the instruments that allow this evolution and that enable this adaptation. I hope that this broadcast enabled you to better understand some of the changes that are upcoming and also to realize the level of preparedness that needs to happen in order to adapt to these changes. We will have further explanations and further discussions on this particular topic in future broadcasts of GFoRSS TV. Once again, thank you to the Waters Corporation for their support in providing the expertise and the time to prepare this broadcast. Thank you, of course, our viewers for your support and your encouragements, and a special thanks to the director of yet another episode of GFoRSS TV, Dr. Eli Bouyezbek. Thank you all, and see you again in another broadcast of GFoRSS TV.
A capacity building initiative such as the one that we are witnessing takes a fair amount of collaboration and preparation. It enjoyed very much the leadership of the Platform of Food Risk Analysis and Regulatory Excellence at Université Laval and the Institute of Nutrition and Functional Food, as well as the Global Food Regulatory Science Society.
So we believe that capacity building is a major activity where we should contribute, and I'm sure the people who are here for training, they will carry a very good experience in terms of theory as well as practical understanding of pesticide residue analysis as well as mycotoxin.
This is a great initiative pertaining to the capacity building. What we are here focusing is basically on the pesticide residues and mycotoxins. In pesticide residues, we are focusing on the multi-residue methods, the polar pesticides as well as the plant growth regulators. Here what we try to achieve is we want to focus right from the sample preparation, separation, identification, confirmation, quantification, data interpretation and verification, which is very vital, or in other words we call it as a total analytical workflow for the residue analysis.
My name is Bazier Raoul Sylvain, coming from Burkina Faso, and I'm working at the National Laboratory for Public Health in the Department of Food Quality Control. This training course is a great opportunity for me to learn more about those chromatographic techniques, especially LCMS or food contaminator analysis.
My name is Kritikam Dong Patrice. I am coming from Cameroon, and I'm working at the National Laboratory for Diagnosis and Quality Control of Agricultural Input and Product, which is under the Ministry of Agriculture and Rural Development of Cameroon. To be part of this training is great opportunities for me, because we who are working on the regulation of policy or concerning agricultural food safety, we are facing a lot of problems, mainly the problem of competencies. Be part of this training, I've learned a lot. I've learned too much. I learned different precautions that you have to take concerning the sample preparation. I also learned how to handle equipment, the sustainability, for its sustainability, and to give accurate results.
My name is GFoRSS. I come from Ghana and I work at Ghana Standards Authority. It's a standard organization that deals with standard development and conformity assessment and also methodology. I'm here to go through this training, that is African Food Safety Laboratory Enhancement Program, which to some of us is very key because it's going to help us to know a lot in the area of pesticide residue analysis and mycotoxin analysis. With a lot of advancement in this area, there is a need for you to be abreast of issues that is going on. So coming here will help me to know the trends in pesticide residue analysis, especially where HPLC is tens of the parts. Right now we are looking at the LCMS. So using the LCMS here will help me to be able to analyze a lot of samples, especially compounds, which is key. With globalization, where export is key for Ghana, there's a need for us to develop a method, use our equipment that we have in a lab to analyze pesticides and mycotoxin.
This training took place this year, and as a demonstration of food safety laboratory testing capacity building programs, it took place at the Dubai Central Laboratory as part of the illustration of what a Center of Excellence of Food Regulatory Science can offer. And in that regard, we're very thankful to the Dubai municipality and its central laboratory for making available the space to us, for also offering the expertise that allowed to set up the equipment.
My name is Lawrence Hello from Kenya. My institution is the National Public Health Laboratory within the Ministry of Health. Now here we are having hands-on approach to the training, which is quite beneficial to us. And whatever we are learning here, whatever the experience we are sharing, we learn from the interaction with our trainers, new ideas. We are even getting new ideas from our colleagues, because you find that the way we are doing things in our place might not be exactly the same as the way our colleagues are doing there.
My name is Daniel Malaya. I come from Malawi and I work with Malawi Bureau of Standards. I'm very happy to be part of this training because we are being trained by experts, experienced in the field, and I find it exciting all the way, always wanting to listen more because we are being taught from first principles, and so it makes it easier to understand.
My name is Esther Mukete Hirunduta from Namibia. I work for the Ministry of Agriculture, Water and Land Reform under the Directorate of Veterinary Services at Center of Veterinary Laboratory, and my responsibility is to test for veterinary drugs residue. We are being taught on how to prepare the instrument for the test, on how to do tuning with the LCMS. We are being trained by the expert from India. The training is quite interesting and we are learning a lot.
That we are teaching them regulation, we are teaching them the basics of chromatography, basics of mass spectrometry, method development, method validation parameters. Then we are providing hands-on practical lessons so that they can prepare samples, they can inject the samples into the instruments and analyze pesticide residues and mycotoxins as per the regulatory guidelines and analytical control criteria.
My name is Jerry Chechetkahu from Nigeria. I work with the National Agency for Food and Drug Administration and Control, which is an organization that deals with the regulation and control of the import and export of food and drugs within the country. I'm very excited to be in this training. I'm part of the participants that have been selected. The training has really been exciting. We've learned a lot on multi-residue analysis using LC-MS.
My name is Kabir Mahmoud. I'm from Nigeria. I work with National Agency for Food and Drug Administration and Control, NAFDAC. I'm deployed to Central Laboratory in Lagos. It's been a great opportunity to be part of this capacity enhancement program for laboratory experts in Africa. It's been a long journey. The program is coming at the right time. The world is such a global village where products are being exported into other countries, and if your analytical process is not in tune, if your analytical process is not detecting what's supposed to detect, there's every likelihood of your product being rejected in the world.
My name is Mugwiza Emmanuel. I'm coming from Rwanda, Food and Drug Authority. I work in a quality control laboratory as food product testing officer. So I'm happy to be in this training because our laboratory is used to subcontract pesticide parameters to other laboratories. So being in this training will equip me with knowledge and skills that I'm going to use when analyzing pesticides.
I am Ido Coco and I am coming from Togo, and specifically from the Agronomic Research Institute of Togo. The trainers are very experienced. They are very good on theoretical and practical level. I have learned many hints during this training that will help me in my future lab works. I found the techniques very good, eco-friendly and cost-effective. What we used to do in 2 or 3 days, we can do it now only in 1 day. It is very interesting.
What we expect, a target outcome of this training is most of the participants should be able to meet all the regulatory requirements for the pesticide residues and mycotoxins.
We of course need funding for capacity building initiatives, and we are very thankful to the US Department of Agriculture, Foreign Agriculture Service, as well as the United States International Development Agency for supporting this initiative throughout its implementation. Corporate partners such as the Waters Corporation and our Biofarm allowed to make available their technologies, their equipment, but also their experts that allowed such implementation. But partner organizations such as the African Union, AOAC International through its different sections, whether be it the Sub-Saharan Africa section, the Arab section, or the India section, were invaluable partners that enabled to make available and to implement this training program.
This training program is affording us the opportunity to keep in line with the latest trend in analytical world. So it's been a great opportunity. We have the best of experts we can find in the world.
I wish we have more and more of collaboration with various other partners and many more.
So it's a good learning experience, and when we leave here, everybody will be enriched in terms of how we do our business back in the laboratories.
I also hope that this initiative will not stop only here, that it will continue to allow us to improve ourselves continuously.
Albeit modest, this training and this capacity building initiative will hopefully represent a major milestone in building the competencies for food laboratory testing in Africa, supporting food control systems and food safety throughout the continent.
The Global Food Regulatory Science Society. GFoRSS mission includes knowledge generation and dissemination, underpinning food regulatory decisions, in support of food control. Our interventions focus on promoting food regulatory scientific disciplines. Consistent reliance on food regulatory sciences enables robust food control programs. Contributing to more resilient food systems and a safe, accessible, abundant and nutritious food supply.
Thank you for watching GFoRSS TV.